Microbial population dynamics in milk. Microbial population in milk is continuously being changed by interacting with the milk components. The initial microbial population of raw milk is being affected by factors such as the equipments and animals' cleanliness, season feed, and also cow's health. The initial microflora of good quality milk consists mainly of organisms found within the udder and of the normal teat skin flora. These microorganisms include thermoduric micrococci and streptococci, which do not grow well at low temperatures. Pathogens will predominate in milk from mastitis cows. Seasonal factors include feed source and housing, both of which influence the level of bacterial spores in milk. Psychotrophic bacteria are low or even at undetectable levels at the best quality fresh raw milk. They originate from the water and from the milk equipments. After collection, the milk is being cooled, which initiates a change in the microbial population from gram-positive rods and cocci, which do not grow, to even gram-negative, which can grow slowly at low temperatures. These gram-negative rods are primarily the Pseudomonas species. When raw milk is not being kept cool, lactic acid bacteria grows, and this results increase in acidity, which inhibit growth of many other microorganisms, including the spore-forming Bacillus and Clostridium species, also some gram-negative Pseudomonas species and coliforms. Growth of lactic acid bacteria produce bacteriocins, which inhibit other lactic acid bacteria as well as some pathogens such as Listeria and E. coli. Both the lactic acid bacteria and the psychotrophic bacteria, which grows in raw milk, are heat sensitive. So when milk is being pasteurized, microorganisms that which have been previously grown in the product are greatly reduced in number. The surviving microflora consists mainly of spore-forming bacteria such as micrococci and lactobacilli. Pasteurized milk, that which is not recontaminated with the microorganisms, can be often kept at refrigerator for about 3 weeks or longer because the surviving microorganisms grow at a very slower rate. Spoilage may occur more rapidly if significant amounts of psychotrophic B series are present in the raw milk. Mostly pasteurized milk is recontaminated with low number of psychotrophic bacteria, resulting in shorter shelf life. Development of closed milking systems, use of bulk tanks to store and transport raw milk, and the improvement in refrigeration systems have caused the microflora naturally present in raw milk to change from predominantly gram-positive producing bacteria to gram-negative psychotrophic microorganisms, which chiefly are the species of genus Pseudomonas. Microorganisms found in milk can also be described according to their optimum temperature required for growth and heat resistance. This is a very practical consideration since milk is being preserved by employing low temperatures to prevent changes due to the microbial activity and also by high temperatures to reduce microbial population and destroy the pathogens. The 4 types of microorganisms are mesophilic, thermoduric, thermophilic, and psychrophilic. Mesophilic bacteria are which grows optimally at 20 to 45 degrees Celsius. The most important mesophilic bacteria are streptococci, lactobacilli, and coliforms. The oxygen requirements can be aerobic and anaerobic. The other microorganisms in this group are micrococci, Corynebacterium species, Mycobacterium species, yeasts, and molds. They are killed in the pasteurization. Thermoduric microorganisms are those which survive pasteurization conditions but do not grow at pasteurization temperature. Most of them do not multiply well at ambient temperature. The most common thermoduric bacteria are found in the genera Microbacterium, Corynebacterium, Micrococcus, Streptococcus, and Bacillus. Excessive number of the thermoduric bacteria in milk makes it difficult to meet the grading standard. Thermophilic bacteria grow well at the temperature used in pasteurization, especially when the low temperature holding method is being followed. These bacteria can grow at 55 degrees Celsius, and most of them have an upper limit of growth at about 70 degrees Celsius. Although some facultative thermophiles may grow at 37 degrees Celsius or lower in standard plate count analysis. The standard method of enumerating this group of organisms is to incubate agar plates at 55 degrees Celsius. Most thermophilic forms are found in two genera, that is, Bacillus and Clostridium. Psychrophilic bacteria are those which are able to grow at refrigerated temperatures regardless of their optimum growth temperature. Usually the term psychrophilic is used for the organisms that which have an optimum temperature below 20 degrees Celsius. In the psychrophilic microflora of milk and milk products, very few are classical psychrophiles and majority of them are mesophilic in nature. They produce a wide variety of spoilage defects. The defects may result in the production of many off flavors and odors. The most commonly encountered psychrophilic bacteria are the members of the genera Pseudomonas, Achromobacter, Vibrio, Flavobacterium, and Alkaligenes. They are killed in the pasteurization process but are sometimes found in pasteurized milk. The contamination takes place after pasteurization from equipment, cans, bottles, and water. Now, looking into the effects of the processing of milk. The principal milk processing operation includes separation and clarification, homogenization, pasteurization, sterilization, chilling, and packing. Contamination with microorganisms is minimal to many thousands, depending upon whether the equipment is properly cleaned and sanitized. Vigorous agitation, such as with the use of pumps, may increase the number of microorganisms in milk by disrupting bacterial clumps into individual cells, each of which can give rise to a colony in plate count procedure. Generally, the water supplies do not contain sufficient chlorine to destroy the gram-negative bacteria. So therefore, the equipments washed with such water must be sanitized before using to control these bacteria. During separation, many microorganisms are physically being removed from the milk and deposited along with the somatic cells and other particulate matter in the slime that contains large number of microorganisms. The heat treatment of milk is the key unit to control the microbial quality of milk. The scientific basis of the specific conditions used for heat treatment of milk and milk products is derived from the concepts of microbial thermal death times, in which the destruction of the organisms is assumed to be dependent on a linear, semi-logarithmic relationship of temperature to time. The overall objectives of the heat treatment applied to market the milk are to eliminate or reduce the risk of disease associated with pathogens common in the raw milk, and to extend the shelf life of milk by inactivation of potential spoilage organisms and degradative enzymes. The specific objectives are to ensure that all particles of the milk are being heated to the desired temperature for the particular time, and to avoid significantly damaging the flavor or other quality characteristics of the milk. Rapid cooling after heating process is also essential to minimize product heat damage and to control the growth of microorganisms which may survive the heat process. In general, The higher the heat treatment, the greater the efficiency or microbial destruction, but also the greater the chance that the quality of the milk may be affected.